Okay, welcome everyone. We can begin now. It's a pleasure and an honor to introduce Professor Frank Doyle to deliver this plenary address at the Congress. Thank you very much, Professor Doyle, for joining us today. I will, on behalf of the organizing committee and on behalf of the Argentine Association of Automatic Control, I would like to thank you very, very much for accepting our invitation. And I don't want to speak for much longer. I will now let Ricardo Sanchez Peña to introduce you. Thank you, Ricardo.
Okay, thank you very much. Thank you, Frank, for accepting this invitation. I've known Frank since the late 80s when we were both PhD students at Caltech. That was a long time ago. Since then, he initiated an amazing career, which included him as a professor at the University of Delaware, at Purdue, at the University of California in Santa Barbara, and currently he's John Paulson Dean of Engineering at Harvard University. There he leads the Doyle group, that is one of the most important leaders in the Artificial Pancreas project around the world. He is fellow of IEEE, IFAC, AAAS, the Association for Medicine and Bioengineering, and he has won several prizes and awards. He has an incredible curriculum. And last but not least, he also even has time to be a soccer referee. So I leave you with Frank.
Great, Ricardo, thank you for that very kind introduction. Let me get my screen sharing initiated. And now hopefully you are able, there we go. Terrific. Well, it's indeed an honor to be here virtually with you at the Congress sponsored by the Asociación Argentina de Control Automático. At the outset, let me start by saying muchísimas gracias por la oportunidad de hablar con ustedes hoy. And I'm afraid that's about as far as I'm going to go in Spanish. It's been 40 años since I studied Spanish, so just a bit rusty. So again, thank you, Ricardo. Great to be here. I'd love to share with this audience some of the excitement around the work that we've been doing, moving from the engineering research domain all the way through to patient care, as I'll show you. So my lab spends a lot of time working on problems in the interface between biology and engineering. And so we, let's see, here we go. So this loop here is often referred to as the virtuous cycle in systems biology. And the idea here is that many of us at the Congress here work on this side of the system, where we do computational modeling, systems analysis, and that informs future experiments in high throughput biology and the unraveling of biophysical networks. We run the full gamut of scales from the genetic scale all the way up to humans and populations. And the idea is that if we think of this as a system, the inputs to the systems are our prior knowledge, hypotheses, literature omics, databases. And the outputs of that system are novel medical insights, whether it's biomarkers for disease, targets for drugs, or therapeutic approaches. So that's the kind of virtuous cycle here. And over the years, as Ricardo mentioned, we've been spending a lot of time in biology. Down at the scale of proteins and networks, we did a lot of work at Santa Barbara around Alzheimer's disease. Also at Santa Barbara, we went all the way up to the extreme end of the spectrum and looking at populations. And we studied a marvelous synchronized spawning phenomena in coral at the scale of kilometers. So really quite a remarkable scale. Another system that we are currently working on is post-traumatic stress disorder, the affliction that affects not only individuals in the military, first responders, but individuals who are afflicted by domestic violence, for example. So we have network analysis to uncover novel biomarkers. Another system we're still currently working on is circadian rhythms, where we study the exquisite mechanism underlying timekeeping in your brain. And this links gene scale all the way up to cells in the beginning of looking at organs. And the last system in my intro here is the one that I'm going to talk about today, and that's diabetes. And for diabetes, we've really concentrated on the scale of cells up through organs to organisms to really understand either targeting strategies in type 2 or the actual deployment of something called the artificial pancreas, which you heard mentioned in the introduction. So let me set the stage why this community in particular should be interested in these kinds of problems. I'm going to reference this beautiful survey paper that was done in Annual Reviews and Control. If you look at the list of co-authors here, they're really the who's who of the control field. They came together 3 years ago and made a projection about the important problems for our field. So if you haven't seen the article, I strongly recommend it to you. One of their sections dealt with healthcare and the notion that most of what we do today, including the treatment for type 1 diabetes, is an open loop form of medication. And the opportunity that lies ahead of us is to move from that open loop mode of delivery to closed loop control. And the financial incentive is massive. At the global scale, they estimate that 50 trillion US dollars, that's trillion with a T, over a 20-year period, so that's about $2 trillion per year, is the opportunity to realize if one can do this. So another way to think about this is consider the body. This is one of my favorite German artists, Fritz Kahn, portraying the body as an industrial factory. And the idea that interventions by control engineers might come in the form of artificial organs. You see a host of examples on the left here. And again, today we're going to talk about the artificial pancreas. Separate from artificial organs, in some cases related, is the idea that you might modulate the delivery of a drug. You might use feedback principles or feed-forward principles to dose accordingly. And there are a host of diseases and maladies that could be addressed through this framework. As I already mentioned, we're going to focus on the artificial pancreas and diabetes. So let me tell you a little bit about the relevance or the importance of that problem. So many have said, and the International Diabetes Federation is the one I'm quoting here, that diabetes is a global emergency. If you look, this particular chart shows you the current state, which is the darker half of these filled in circles at the bottom, region by region, and in this blue circle, the entire world. The upper half of those circles denote the forecast in 2045 of where we will be. And I'll call to your attention that locations like Africa are expected to increase 150%, Middle East and North Africa over 100%. It's not quite as dramatic, but in South America, 60% increase over that time frame. So this really is a striking problem. Financially, it's very consistent with the numbers I showed you earlier. Altogether, year over year, it's about a 700 billion, that's billion with a B, US dollar worldwide cost for the associated healthcare expenditures connected to diabetes. And you can see that just in North America and South America alone, that's 400 million of that. US, North America is about half of the total cost. So huge opportunity to improve the treatment of diabetes with a dramatic financial opportunity. I should point out that they're in Argentina. I wish I could say here in Argentina. I wish I was with you in person, but there in Argentina, the burden of diabetes is quite profound and has been studied in a number of papers. This particular paper just came out just under 2 weeks ago, reporting on a global analysis of disease burden. And in particular, I focused in on the country profile for Argentina. What you're seeing here are the 10 highest causes of death and disability combined. And they're measuring this. The metric they're using to evaluate this is the disability adjusted life years. So if that is shortened by the disease, that becomes a problem, of course. You'll note that diabetes is number 5 on this list. It is the one that has increased most dramatically over the last decade. So a serious problem worldwide and definitely a problem there in Argentina as well. Okay, so I'm going to walk you through some of the basics of what's going on in the body with glucose regulation. With apologies, I know Ricardo knows this very well. Some other members of the audience may know this. If there's any bioengineers or medically trained folks, forgive me for a quick tutorial here. This is a very simple sort of schematic of the feedback control underlying healthy glucose regulation. You could think of your pancreas as a multivariable controller. Primarily, it modulates the secretion of two hormones, glucagon and insulin. We focus on insulin initially when your blood sugar is lowered. So for example, after exercise, blood sugar goes down, signal to the pancreas to then secrete glucagon. Glucagon stimulates, enzymatically catalyzes the breakdown of glycogen in the liver to form glucose, and that leads to an elevated blood glucose level. On the other hand, when your blood sugars go up, so there in Argentina, also here in Boston, it's just after the midday meal. So many of you had some carbohydrates. This caused your blood sugar to elevate. That stimulated insulin secretion by the pancreas. And insulin is like the brakes. It enables the uptake of glucose in your muscle and fat cells. And that, of course, will lead to a lowering of blood sugars. So in some ways, it's like an analog to driving a car, where insulin is the brakes and glucagon is the accelerator. That's the sort of dual role that these two hormones serve in a healthy individual. And here's an example of what happens with a healthy individual. This is actually one of my PhD students from University of California back in Santa Barbara. He wore a glucose sensor for a 24-hour period. I draw your attention to the axis here, 0 to 400 milligrams per deciliter. Throughout the talk, I'm going to use the same axes. So in your mind's eye, you can always reference the same scale for consistency. So what you see here is, in effect, a remarkably healthy flat line, very, very tight control. And the thing that intrigued me after this study was, or data was collected, that grad student shared with me that it was a Friday. And since he was a graduate student, his normal Friday involved pizza, involved beer, and involved late night bowling. So despite the fact that he was subjecting his body to these disturbances, we would call them in the control space, you can see the controller in his body was controlling his blood sugar remarkably. Now, in the case with somebody who has type 1 diabetes, the pancreas no longer makes insulin. So I've removed that part of the feedback circuit here. The immune system attacks those cells in the pancreas that make insulin, and that's no longer functioning. There can be some compromise of glucagon production, but the primary effect is insulin is no longer produced. When that happens, of course, your blood sugars escalate. We refer to this as hyperglycemia. And hyperglycemia is dangerous over the long term. Hyperglycemia leads to compromise in the tissues and the organs everywhere where the blood flows. So you can imagine all of the manifestations of that as sort of sketched out in this figure here. So what happens? People who have type 1 diabetes, the standard protocol, not the high-tech solution, but the standard would be to put a human in the loop. Have an individual check their blood sugars, do a finger stick, run some calculations in their head or on a calculator or on the spreadsheet, and then manually dose their insulin. Now, I don't need to tell people attending this Congress the fact that putting a human in the loop is a bad idea. We want automatic feedback control. So when you put a human in the loop, they can overdo it. They can have errors, they can have uncertainties. If they put too much insulin, now we run the risk of the very dangerous condition of hypoglycemia. Hypo in this case means it's lowered, it's brought down potentially to a dangerous level. And what you see is the increasing severity, depending how low the blood sugars go, from just headaches, confusion, ultimately coma, and in the worst case, death. So clearly, this is not what we want. And a person manually managing their blood sugars will have very erratic control. So again, here's a plot, same y-axis. In this case, the x-axis is 1 month. This is one of our patients from Santa Barbara. And I deliberately picked a subject who was, shall we say, middle of the road. They weren't the most disciplined and effective manager of their blood sugars, but they also weren't one of the worst. They were somewhere in the middle. This green zone that you see highlighted here, 80 to 180 milligrams per deciliter, that's the zone that doctors would like to keep their subjects in to minimize side effects. So if we're able to keep the subject for a high fraction of time in that zone, they're doing pretty good. What you see with this subject is there were a lot of hyperglycemic excursions above that zone. There were a number of hypoglycemic excursions below the green zone. And this red line at 50 is a literal red line. That's a very dangerous threshold. And you can see there were close to a dozen events that were happening during this month-long period. So I'm going to give you a summary of these results, and I'd ask you to commit this number to your memory for later in the talk. This typical subject managing their sugars on their own, so this is the open loop base case, is able to stay in the zone 55% of the time. Use that as your mental baseline. They're elevated about a quarter of the time, and they are depressed below 80, hypoglycemic, about 20% of the time. So kind of keep that in the back of your mind as we talk about alternative strategies. So if we go back to our diagram, again, the key here, get the human out of the loop, put something automated in there. And so we've been calling the system that we've come up with the APS, the Artificial Pancreas System. We started on a laptop in a clinic. We moved to a tablet that could be carried around. These days, it's an app on a phone. And we've got a couple dozen subjects right now running this on their phones in a clinical trial. This is just to show you a bit of a roadmap. It's been a really interesting 100-year journey to get us to where we are now. So if you go all the way back to when Banting and Best first administered insulin, the first place they did that was in Canada. The second place in the world where they administered insulin was actually Santa Barbara, California, my former home here. And over time, there were improvements in technology, although they were kind of cumbersome. If you can make out this picture, this is the biostatter. This is the size of a large bookshelf, much bigger even than a modern computer. That was the state of the tech, and it was a bedside solution. As you moved through, you begin to see where about 15 years ago, the Artificial Pancreas project was launched, and Ricardo mentioned this at the outset. This was the beginning in the US of a major funded initiative, and I have to say, to the credit of the JDRF, they made it an international initiative and funded folks from all around the globe. The NIH got involved as well. And about the next decade or decade plus were the exciting developments in that front. We were excited to not only close the very first feedback loop in the US in the summer of 2007. Here's the picture showing that. But then we went and developed a number of other firsts. We were the first outpatient study. We were the first app. So we've got a number of things that we were able to be first in. And if I take us right up until 1 year ago, we were privileged to be part of a larger study that was being led out of UVA. We were the algorithm team. UVA was leading the overall study that got published about 1 year ago, in fact, almost exactly 1 year ago, in the very prestigious New England Journal of Medicine. So what's the state of the tech? Many of you out there in the audience are controls engineers, you're algorithm types. Here's the technology we're dealing with. These are the actuators and the sensors for our feedback system. So these days, there are very, very efficient sensors. They're very tiny that report out through telemetry, Bluetooth, every 5 minutes. And there are pumps that can now, I can say this, a year ago I couldn't say this, that actually are in the market. They're able to do closed loop control by connecting sensors to pumps. So we're just beginning to see the first wave of this. In the US, the fraction of people with type 1 diabetes that are on a pump is still only about 40%. It's a relatively low penetration of that technology. I don't know the figures for Argentina, but I'd be willing to guess it's probably less than 40%. Maybe Ricardo can update us in the Q&A session. So I'm going to walk you through two sections of my talk. The first is just a quick snapshot of some trials we've done. But then I want to dig deeper on algorithms, and in particular, the most recent algorithm we're working on, which is currently being tested in a clinical trial. So here's the road that we have traveled for the last dozen years. What you see in this plot are the number of subjects that we've had enrolled. It's a cumulative plot. In the back, what you see are the number of hours of closed loop on the technology that's come out of our labs. So we're closing in on 1,000 subjects, and we've logged about 200,000 hours of closed loop patient time. The very first closed loop I mentioned earlier that we did, summer of 2007, handful of subjects for just a few hours each. The most recent study that we wrapped up was 6 months in duration at home. So we really have come a long way, which is why in my talk title, I refer to patient care. We've really gotten to the point of that. So let me give you a peek inside one of these trials. By the way, I should point out, of course, in early 2020, when COVID hit us and you and the world, we slowed down trials. In fact, we stopped trials in March. So I haven't updated the slide because things have been put in a frozen state for about the last 6 months. We're just resuming the trials now. So here's a trial that we had just wrapped up before COVID shut us down. This was a pediatric study, so very young children that we were looking at in collaboration with colleagues at Yale, also with the Sansom Diabetes Center and a couple of other sites. So here's the idea. We were going to have these kids go through a 3-day protocol, starting with lunch at the beginning, group activities, dinner. And I should say that these were completely unconstrained meal and activity protocols. We would take these kids to a playground and let them go loose. We would put them in a restaurant and they would eat whatever they wanted. So this was really a great test of the tech, if you will. Here are some of the example results that we got. Again, exercise was not programmed or announced. The algorithm knew nothing about it. And there were no restrictions on meals. And as you can probably see from the picture here, some of these kids went a little crazy with their meals during the clinical study. You see here the devices that they're running their closed loop system on. By the way, the young kids in particular, really fascinated with the tech. Love the idea that this is almost like a game on their phone and not some new burden or responsibility they had to assume. So what you're seeing here is comparisons of open loop and closed loop. And what you're looking at is meal size. And what you see is in the closed loop trial, these kids increased the amount of food they ate. So really it was a significant challenge to the closed loop system. So here are some of the activities. I don't know if this video will play now. I had a video plugged in. It doesn't seem to be working in this system. But this was a video of a kid jumping on this bouncy thing, eating food, climbing. It was really a test of robustness, right? So here are some scatter diagrams showing you, first on the left, mean glucose. On the right, the time in range. So that number I told you, which was 55% for a relatively disciplined manual adult. What you see is these kids were all over the map from 25 to 80, average about 57. We improved it, even with the increased food load, even with the crazy activities. We still brought the average time in range up, and we lowered the average blood sugar. And these are color-coded by the ages of the individuals, the adolescents versus the children versus the very young children that we had in the study. So it gives you a sense of some of the excitement when your tech can have an impact on a community like this, young children. It's very, very exciting. Okay, so now let me dig into the algorithm, probably the part that many of you in the Congress here would be most interested in. So I'm just going to walk you through some of the basics of how we approach this problem. And if I go back, it turns out that our very first paper on this was nearly 25 years ago. This was a paper at a IEEE, Engineering and Medicine and Biology conference, 1996, where we proposed for the first time that model predictive control would be the right architecture to use for this. I'm sure people in the audience are quite familiar. I'll just quickly recap for you. The idea is we have our subject with the attendant stresses that I've been referring to, meals, exercise, other stresses. We have a continuous glucose measurement device, which requires periodic calibration. That repeats at a 5-minute rate back to the controller, the blood sugar number. And within MPC, we have a prediction model of the subject. We use a time series empirical model to make a forecast in the future. Formulate as an optimization problem then the insulin trajectory in the future that minimizes a certain cost function subject to the constraints that we impose, and I'll speak more to that. We solve that through an optimization problem. So that's the basics of the MPC approach. So what we've done over the years is we've taken the basic algorithm and tried to customize it for the individual. One of the things about medical problems is no two people have the same manifestation of a disease or a malady. They really require tailored, customized solutions. So we've done that a couple of ways. One way is to get the right patient attributes, build this into a model or a parameter in the controller that's peculiar to the patient, something like the basal rate, the amount of insulin that's delivered separate from the meals, or the carb ratio, the multiplier that you use to cancel the effect of a meal. Think of it as like a feed-forward gain on the disturbance. Another category would be to adjust the tuning of the controller. To now use a strategy where the information about the individual informs a setting in the controller, whether it is a penalty in the MPC, whether it's something like an extremum control strategy, or whether it's a velocity-dependent adjustment of the penalty function. Then the work that we've done most recently, and the work I'll talk about in depth, is really a combination of both, where we both adapt the patient's parameters and the controller tuning, so basal rates, carb ratios, and the controller tuning. And that's the work which is underway as we speak. I won't be able to show you clinical results because those subjects are literally in the middle of their trial, but I'm going to walk through the algorithm with you and some of the in silico work. So the beginning of our work actually goes back to 2002 when I moved to Santa Barbara, and I met this brilliant doctor, the late Yost Jovanovic, and we together worked on the idea of trying to do adaptation inspired by run-to-run control. And we continued that work and did clinical studies. This is Dr. Howard Zisser, who was at SANS at the time. Today, Howard works for Google. The idea was the following. We borrowed from traditional engineering controls where repetition was key. So I'm a chemical engineer by training, so the semiconductor manufacturing or batch chemical processing. For those of you that are in the robotics space, robotics is another great example. When you have a repetitive process, time and time again, you don't know necessarily how well you're doing until the final part of the process. So in the case of batch chemical processing, what the final produced chemical attributes are, then you know how well you did. So there's a whole host of algorithms, iterative learning control, run-to-run control in that space. Here's how it might look for diabetes. This is how we looked at it for diabetes. You could take your 3 meal-related boluses, the cancellation of your meal sugars, and you could adjust their timing and their amount based on how you did in the previous cycle. So for diabetes, 24 hours is a great cycle. For many of us, we eat 3 meals a day, day after day. We have them timed at relatively similar times, day after day. So those are patterns. Those are recurrent patterns we can try to fine tune and optimize. So we studied this, and the idea was if you started a subject well out of control up here, I apologize, this isn't the same 0 to 400, this is from 50 to 300. But if the subject's out of control, each of these spikes are the equivalent of a day. It's the peak over a day. What you're seeing is over about 10 days to 2 weeks, you can pull that subject in using a run-to-run control strategy like you see here. So that's an example of doing run-to-run control. This is actually the first closed-loop trial we did with frequent measurements, 5-minute sampling, using a model predictive control algorithm where here we tried to customize the individual's parameters. So here's an example of the patient. What you see, we published this back in 2013. This was the study that initiated back in the summer of 2007. We were able to get in range 70% of the time. So if you remember, back at the beginning of the talk, I told you keep 55% in your head. For our first closed loop study, we would have been happy with 55% because we've automated the delivery. We've taken that burden away from the subject. So we can reproduce what they're doing automatically. That's a win. So we not only automated the delivery, but we improved their time in range, which you would expect with an automated system. You notice where we made the big headway is in the hypoglycemic. We have virtually no hypoglycemia. We still have a good deal of hyperglycemia, but we've consolidated time in range as a result of this algorithm. Our study that followed that one was slightly longer. This was a 27-hour study, just slightly more than a day. Slight improvement in the time in range, now to 75. Overnight, we're getting dramatic control. Now, let me be clear. Overnight is, on one hand, one of the easiest times to deal with. There's no exercise, there's no meals, the subject is sedentary. But overnight is the most fragile time. If the individual is dipping low, they're often unaware, they're sleeping. And so the idea is that we really, especially with children, have to have tight control overnight. So in only our second study, we were able to get to a 99% level overnight for these subjects. These were adults. So let me tell you a little bit more about the algorithm. Here's our what we call zone MPC algorithm. So we are penalizing for deviations outside of a zone. So much of this should look pretty similar to those of you that work in the MPC space. We have a error term that we're seeking to minimize by calculating future insulin delivery. And here we have two cost penalties on the insulin, one for positive excursions, one for negative excursions. So I'll explain why that asymmetry is so important. Down here, we have the usual constraints, follow the patient model. It's a state space realized model here. We calculate excursions from a zone. I'll show you that in a pictorial manner in a second. Then we look separately at positive and negative insulin deviations from a basal baseline value. So we calculate that future series, we implement the first one, and we march through and repeat the procedure using a receding horizon control strategy. So in a sort of pictorial form, here's some of the attributes of this that make this not a generic MPC problem. So first of all, we bound the glucose, we create a zone, and anywhere inside this zone is given a zero cost function value. So we only penalize when there are excursions outside the zone. Part of that is because we don't want to chase noise in the sensor. Part of that is because the doctors prescribe a range of values that minimize side effects and complications. You don't have a magic single value or even a value that varies over the day. They talk about ranges in the medical profession, so we should capture a range in our control design. Same thing can be applied to the pump. We have obviously lower bounds. We can't take insulin back out of the body. We have upper bounds, which are either mechanical or safety limited. And during certain periods of the day, like overnight, we could even tighten those bounds to limit how much activity will happen during closed loop control overnight. We have safety limits also on the pump, which dictate how much insulin we allow to circulate in the body. It turns out insulin can remain active and circulating for several hours in the body, depending upon the individual's body chemistry. So we don't want to keep loading the insulin, something called stacking the insulin, as we've already delivered by adding more. So that's a safety override we put on the pump as well. And finally, we've got these asymmetric performance ways. I told you at the outset of the talk, hyperglycemia is the long-term complication of diabetes. We measure that over months and years. Hypoglycemia is immediately dangerous. We're doing things like starving the brain of fuel. So that's one we have to act immediately. So in terms of deviations around the basal, we have very different consequences to going high versus going low. So this version of the algorithm was one that we tested just a couple of years ago. This became our long 12-week study. So this was a 3-month-long study. We had, in this case, the adults who completed this study, there were 30 of them, had a target, a goal of the A1C. Now, I don't have time to go into all the details, but the A1C is a measure of glucose. It's the glycosylated hemoglobin. And that is the gold standard for how well the insulin, how well the subject is managing their diabetes. What you see is we took subjects who were actually in a very good, you'll have to trust me that 7 is a very good number for the A1Cs. And even though they were a disciplined cohort, they had a statistically significant reduction of their A1Cs. Part of that corresponded to the fact that they had a lot of hypos in the open loop. SAP means sensor augmented pump. It's manual control, open loop control. Under the AP, closed loop, we got down to the numbers that I reported earlier. So we had dramatic improvement there as well. Now, one of the most recent adaptation algorithms we've looked at actually was the last closed loop trial that we have published so far. As I mentioned, we've got one running right now, but this one used a novel form of adaptation. We referred to it by either a trust index or confidence index. So let me give you a sort of a cartoon to try to explain this. This is a forecast of the S&P, the stock market. It was taken back in late 2017. And what you see are these error clouds projected out in the future. Any of the members of the audience who are sort of my age or older will be very familiar with these. These are what your retirement portfolios give you when you try to calculate what your future will look like. These are the error bars. These are the projection accuracy going forward. By the way, if you look at this, because this was done in late 2017, we're now late 2020, the forecast was that we'd be around 2,500 for the market. The market's actually 3,300 right now, the S&P, although I think it went down quite a bit yesterday. So it was right at the edge of the white cloud here. But the point is that we know that there's uncertainty. We also know that the market volatility is actually a measure. There's an index you can track online, the volatility index, that tells you when it's a sort of risky time for stocks. And this is a 5-year snapshot. And what you see is actually it's been relatively quiet during most of the last 4 years in the US. And in this COVID period, we've had this spike. If I zoom in, this is the year-to-date projection of the volatility index. You can see a huge spike here. What's the relevance to control? We want to map confidence into the controller aggressiveness. So for example, if we're in a range of behavior where the patient is tracking the model, where the sensor is highly accurate, where our predictions are highly accurate, we can be more aggressive with control. But when the variability increases, whether it is sensor noise confounding things, whether the model and the patient begin to deviate more, then we back off. So that's the idea of this trust index that we developed. And here's how it would play out in the algorithm. Those weights on the insulin delivery, the positive versus the negative delivery, now will be adjusted. And if I'm in a high trust mode, if I'm in a mode where I believe the sensor, I believe the model, I have a lot of confidence, my subject goes low, hyperglycemia, controller, I'm sorry, they go high, hyperglycemia, the controller will aggressively deliver insulin. If on the other hand, my trust index is low, noisy sensor, bad model, whatever, we're going to dial back on that aggressiveness. We're going to be less aggressive on that delivery. And you could do the same corresponding analysis on hypoglycemia. When the subject goes low, if I'm in an aggressive mode, I'll immediately turn off the pump, reduce that delivery, dial it back in a fairly reasonable way. If I'm in a conservative mode, I don't trust things, subject looks like they're going low, I'll be more conservative and I'll shut down even faster that pump because I don't want to be wrong. So that's how this would play out. And when we implemented this in silico, we got some very encouraging results here. Sorry, this is the clinical study. Silico was great, but of course in silico is on the computer. So out of MATLAB, we go to an actual subject trial. What we had here was 2 overnight periods. They came in before dinner, overnight, exercise or walk, have some, again, meal, overnight period, lunch. We ended the closed loop right before dinner then on that 3rd day. So we had 15 subjects. And you can see we got a remarkable 88% time in range. This is 24/7. This is not overnight. This is 24/7. A very, very low 1.5% time below 70. Some summary of those results are given here. Here you see the average blood sugar for those 15 subjects. And the thin lines above and below are their interquartile intervals. You also see the dramatic improvement. I showed you a plot like this with the children from the run-in week, the beginning of things, to the closed loop session. You can see the dramatic improvement in the cohort up to that average of 88% time in range. So this was the last fully closed loop trial published using adaptation. And just to put it in context, I'm going to show you a map that we've been reporting in all of our studies. We've done a deep review of the literature, and this is 6 years of everybody's results. Anybody who has published in the open literature a clinical closed loop. So again, all the studies out there, if they're closed loop and with a real human, clinic or at home, they show up in this chart. And what you see For example, that last study that I just showed you is this purple circle up here. This is the proverbial high watermark so far in published clinical work with an 88% time in range. Clearly where we want to be is in this upper left corner. Time in range very high, time below 70 very low. So the ideal spot would be 100% of the time in range with no lows. And you can see there's almost a Pareto curve here where there's some trade-offs with getting very, very high, but not necessarily being able to reduce the number of lows. By the way, the bigger circles you see down here were some of the pivotal and pre-pivotal trials for the commercial devices that are out there, such as this one here. So it gives you a sense of where the margin for improvement might be with these devices. Okay, so I'm going to bring you here at the end of my talk into our last algorithm, the one that's currently being vetted. And this was a study Dawe Shi, who was a postdoc in my lab, did with A.L. Desau, a longtime member of my lab, and myself. We published just last year. It was a classic adaptation algorithm. So we had a core inner loop that used the MPC design that I've been describing to you. And then we had a slower, longer adaptation part of the loop. This would occur on a weekly or bi-weekly basis to fine-tune controller and patient settings in the inner loop here. So lower loop, the MPC calculation, upper loop, chronic changes, whether it's lifestyle or metabolism of the individual, based on data we collect, we can then adapt and refine the parameters in the lower level algorithm. So here's a flow chart that shows you the logic underlying the adaptation. So the subjects start out week 0. We do some first 2 iterations about basal rates. Depending upon if the zone is achieved, we'll continue on to look at control parameter. If it's not, we'll do a carb ratio adaptation, maximum up to 5 times, then go back and check basals. If we pass through here, we do a control parameter update. There might be an iteration on that as well until we converge and come out at the end of the study. So this is the idea. We're using a Bayesian optimization approach in this, and I can speak to that more detail or refer you to the paper for some of the algorithmic details. Here's an example, though, of how we do one of the adaptations. I'm just showing you one of them here. This is the carb ratio update. So this is a meal-dependent glucose management objective. Depending how we come out of the meals, what the post-meal or postprandial profile looks like, we'll use by way of information to adapt or update the carb ratio. So what you see here is the optimization we're trying to achieve to guarantee that we settle in a relatively quick way down to the 120 to 150 range after a meal. Some of the constraints that we impose on that optimization: first, we don't want to change the carb ratio too quickly between adaptation updates, standard sort of adaptation throttling, if you will. We have a bound on the average blood sugars that we want to achieve through any adjustments. The bounds on the carb ratio itself, we have some sense of the physiology. And then we also have a hypo-safety constraint that we want to stay above some minimum carb ratio so that we don't induce a hypoglycemic event. So that's how this might look like in the code itself. We're using a Bayesian optimization to get the profiles that we would use to minimize this cost function. So what does it look like if we put it in silico? Here are some profiles where you can see the system was started from a condition. On the left, the blue is time in range. You see that marching up over the course of weeks. On the right-hand axis, and in red here, is the mean blood glucose level. You see that lowering as we get stabilized control. And down below here, you see where there might be either time below 70, of which there are a couple bumps, or time below 54. That's the red line, for which there are virtually none here. So we got a very robust learning algorithm in terms of the results that we were able to achieve and publish in that paper last year. We started our subjects with underestimated basals and overestimated insulin to carb ratios. We start out 41% in time. To an adaptation algorithm, we get up to 88% time in range. If we do a variation on this that's even more difficult in some respects, overestimate the basal and overestimate insulin to carb, we might appear to start out in a good range, but now we have to unwrap these conflicting demands. And here we actually got up to 88.8 time in range, statistically significant. and took away what began as dangerous hypos and completely eliminated them. So this had great promise, and this is the algorithm that's currently being tested with subjects in Stanford, Palo Alto, in Santa Barbara, in Boston, and at the Mayo Clinic. For these studies, we actually are running things on a phone. I showed you the children and the phones that they had there. This is the ecosystem, if you will, that we use for putting an app on the phone that has our algorithm embedded. We can also use that app then to project up to the cloud the data, so the doctors have real-time glimpse into the trials and can study them and monitor them. I can access through a web interface all of our current subjects and what their trajectories have been. So a lot of experience with that, a lot of work went into that. This was really the brainchild of A.L. Dessau in my lab, and has become the workhorse of our clinical studies. So as I'm wrapping up, the obvious question is, what's the future look like? Certainly plenty that still needs to be done on this system. One thing I'm a big fan of is thinking about embedded design for this. So we have spent some time thinking about how we would put a chip on a sensor or a chip in a pump. We would actually embed all of the system, so the control unit as well as the data acquisition unit, in an embedded design. And we've done some preliminary work to prototype this and show how this might work. Not only do you get a very convenient and robust footprint, you also can achieve some energy savings depending upon the way you design this. I think another way to think about the future is that this is going to be part of the growing world of the Internet of Things, particularly in the medical space. If we think about all of the things that we all carry on our body and our phone, cameras, you could take pictures of food, your calendar, you could track your exercise time, your meal time, your activities, lots and lots of things that could be accessed and fed into an IoT-based system to do those calculations. It would be patient-centric, but the docs, the engineers, everybody would be in the loop to be able to help support a system like that. So this suggests immediately a couple of different things that we might build into a future AP. We've done preliminary work on exercise to understand the impact of exercise. Exercise is a hard one that I could talk about in the Q&A. Very difficult to try to project whether it's going to be an up or a down signal. Meal bolusing, we've done some preliminary work here using things like machine learning approaches to analyze food images and then estimate nutrient content, to then fine-tune the insulin delivery. And many other ideas around personalization using prior health records or long-term accumulated records to inform updates and refinements. Something else that's an ongoing study we've got right now in collaboration with Joseph Wang at the University of California, San Diego, are additional metabolic sensors. So some of you out there may be saying, okay, you've told me a story with one sensor, glucose, and one actuator, insulin. How hard could that be? I hope I convinced you it was a pretty hard problem, but one could make a more sophisticated solution if I had more information, better sensors, for example. So I'm showing you here that Joseph in his lab is putting together exquisite sensors, some of which are multi-marker devices where you can have on one sensor a multiplexed array of sensors for things like lactate, cortisol, insulin. Turns out to be a really interesting measurement to include, to know that one would stay away from the dangerous boundaries, for example. So I believe these are going to be game changers. They're not ready for the market yet. They're not ready for a subject to plug and play, but it is the beginning of getting new signals into the AP space. Okay, so final thing I want to say, I've had the wonderful pleasure of speaking with you this afternoon and talking about the work, but these are the folks who have done all the hard work over the years from my lab. The names in white are the graduates who have left the lab and gone on to industry, academia, graduate school, wherever they might have gone. The names in blue here near the bottom, or teal, are the folks currently working in the lab, Sunil, Bashak, and Eleonora. Over the years, I've had wonderful, wonderful medical collaborators. So Dr. Zisser, starting at Sansom and now at Google. We have a closed loop trial study group called the IAP. It's an international collaboration you see here. And the work I reported on the adaptive control was part of a NIH DP3 grant with the consortium you see here. These folks pay the bills. These folks provide equipment and tech that we can use in our trials. But my final slide I want to reserve for the most important people in the whole work. And those are the subjects. So I want to let you hear in their words their appreciation for what's going on.
The artificial pancreas is phenomenal because it will allow me, if I accidentally fall asleep early, to monitor my blood sugars throughout the night without me having to worry and get up. It'll mean when I'm working out and I'm dipping low, it'd be turning off my insulin. There's so many advantages with having a machine that's constantly monitoring you than you taking a break from your day and just keep testing and keep adjusting when the machine's helping you do it.
So there you hear it right in their own words, and this is what motivates and inspires us day after day to do this important work. So that completes my remarks. Again, I want to thank you for the invitation to talk about this work with you today. And Ricardo, I'll go back to you and learn how you want to run the Q&A discussion. Be happy to answer some questions.
Okay, thank you. I'll do a virtual clap. Thank you very much, Frank.
Thank you very much.
Very interesting. I will leave the Q&A to the conference organizers, to Hernan, probably.
Okay, thank you very much, Frank. It was an amazing talk, very interesting. Yes, any questions from the audience? Please raise your hands. Okay, we have many. Just let me. Okay, I will. Fabrizio Garelli. Yes. You can activate your microphone.
Sorry. Hello, everybody.
Hello.
Thank you very much, Frank, for your talk. It was very amazing. You wonder about the pump or the technology penetration in Argentina. I'm afraid the pump penetration is just about 1% here. At least, as far as I know, that was the last year we had. And now a question. I would like to know, what do you think about the bi-hormonal artificial pancreas? I have seen very impressive results, but they make hardware quite more complex. So I would like to know, what do you believe, if they are actually necessary to accomplish good glycemic control, or if you believe they're, I don't know, more necessary as safety system maybe?
Well, thank you, Fabrizio, for your question and for the data point about pump penetration. I should point out that even with injections, there's technology you can bring to bear that approaches closed loop control, but at a very slow time scale, of course. So in terms of the bihormonal pumps, this is a great point. I didn't really mention that earlier. Thank you for asking. There are some groups around the world who are looking at glucagon and insulin to deliver in that counter-regulatory kind of arrangement that I had mentioned. And so here's what I think about that, Fabrizio. First of all, today there is not a stable formulation of glucagon at body temperature. So this is the first problem. It's not feasible today because we don't have a stable version the way we have stable insulin that can stay in a pump and not aggregate. Smart people are going to figure that out, though. The chemists are working hard on that. The second thing we don't know, unlike insulin, where we have 100 years of studying this, we don't know what the constant pulsing of glucagon on the liver is likely to lead to in the long term. So if this is going to be a problem that over time you actually degrade the quality of glycogen or glycogen breakdown, that could be a problem, and we don't understand the medical dimension. Now, that would all be fine. I believe that we'll get the knowledge on the medicine side, and I believe that the chemists will figure out a good, stable, crystalline structure for glucagon. Where I have more concern is there have been studies, and these were done in Canada about 2 or 3 years ago, benchmarking in a very fair way single hormone versus dual hormone. And what they reported in those studies were indistinguishable improvements. So the two benchmarked very, very tightly. And so to the point you raised in your own question, the added complexity of adding glucagon does make this a more complex system. Is it worth that if you're not actually improving closed loop control? So that, to me, it's an open question. We haven't studied this in the lab. There are smart people working on this, and one day we may get there because, after all, that's what our body does naturally. But I think the jury is still out today on how well motivated that is.
Okay, thank you very much.
Okay, next question, Alejandra Ferreira de Loza.
Hi, good morning. Thanks for your interesting talk. I would like to ask you, how do you compute the reference signal? I saw a reference signal in one of your slides in the beginning. So I'm interested in know how do you compute such a reference?
Yeah, thank you, Alejandra. That's a great question. What we use is a zone for the reference trajectory. And so maybe I can go back. Let me see if I can pull this up quickly here. Here we go. Hopefully you can see that on the screen now. So our reference is actually a zone. It's not a single value. So what we feed into the controller are an upper and lower bound that change over the 24-hour day. For the first trial that we did, we actually elevated overnight the sugars so that we would be doing less aggressive insulin action. That was required by the FDA for us to get approvals. So depending on the trial, depending on the subjects, we may have different widths to these bounds. We may have different elevations at night, or we may be constant overnight. But this is that reference input. It's actually a zone that we feed to the controller.
Okay, thank you. Just one more question. This zone is individualized, or is you have a set of several zones?
So we have looked at in silico using MATLAB, customizing the zones. We haven't done this in a clinical trial yet. And there certainly are some people that would have different zone characteristics and it would make sense for them. But I haven't tested that in the clinic yet. So I would say there's encouraging theoretical results. But in clinic, we have made the zones the same for everybody.
Okay, thank you.
You're welcome.
Okay, next question from Hernande Batista.
Hi, Frank. How are you doing?
Good, thank you.
My question is, I would like to know your opinion about the new concept of interoperable insulin pumps. You know, there are lots of control algorithms being developed worldwide. Do you think interoperable insulin pumps will help patients to get access to control algorithms more suitable for them?
Yeah, so great question, Hernan, and I'm a big fan of that. For people who don't know, it's interoperable approvals are that the agencies are agreeing that the devices being put out, like sensors, like algorithms, like pumps, don't need to be reevaluated every time a different combination is tried, but there are approvals given to be interoperable. So I'm a huge fan of that. In fact, that APS system I described for our work, we've always been device agnostic. We've always said we'll work with whatever sensor, whatever pump. And so from the very beginning, my own platform, APS, was an interoperable platform. So we're a huge believer in that. I think implicit in your question, though, was a sort of a question about these new DIY approaches to doing this. So people may be aware that there's a huge community of individuals with type 1 diabetes who are designing their own algorithms. They're running their own spreadsheets, and now they're sharing those algorithms. But the idea that a do-it-yourself approach would make sense. I have to say, there I'm more cautious. because there's tremendous value in having a regulatory agency review a protocol, review an algorithm, ensure that it's safe under a wide range of scenarios. So for individuals to bypass that safety review gives me great concern. And there already have been stories and incidents reported in the press about individuals who had dangerous hypoglycemic excursions from DIY algorithms. So I love the idea that the community will help foster innovation. We have to think about how to reconcile the regulatory process along with these creative do-it-yourself helpers.
Thank you, Frank.
My pleasure.
Okay, next question from Ricardo Sanchez Peña, who is very, very impatient to ask you a question.
Okay. Okay, thank you very much. Again, as usual, you made a very clear talk, so thank you very much for that. I have a short question. When you mentioned about perturbations to the system, like meals, exercise, and stress, what type of sensors do you use for stress as a perturbation?
Yeah, so the quick answer is today we don't have one, right? And so the idea, if I go back to my, let's see, if I put this slide back up. So the idea would be, for example, lactate or cortisol would show up whether you had a physical stress, you did an intense workout. You might see cortisol surging if you just have a kind of a fright or anxiety stressor. So one of the challenges here is the stress you might get from anxiety. Let's say you're a student and tomorrow you have exams. That's one type of stress. Let's say you're an individual and boy, I don't want to say COVID, but you have the flu, you have the common flu. Your body is going to have a stress response. And then on the other hand, let's take somebody who just ran a 5-kilometer road race. All of those individuals will be manifesting different stress profiles. Some of them we might be able to distinguish by looking at lactate versus cortisol. Some we might be able to distinguish by looking at actigraphy. So for example, if they have a watch with an accelerometer, you can pull out that guy who just did the 5K race. That one's relatively straightforward. So I would say the answer to your question is the next generation is getting those stress markers. The only thing we've been able to do so far is we have actually induced stress, which for a moment might sound counterintuitive, but we wanted to titrate how much stress led to a change in the insulin requirements. So we gave subjects prednisone, which is a steroid. In fact, it's a steroid that doctors will often prescribe for very bad allergies. That modifies the insulin sensitivity. So we have titrated subjects with prednisone. That was our stress proxy to understand how much the stress led to a change in the insulin requirements. So we've got some of that understanding, but we really need what's on this slide. We need my good friend Joseph Wang and his team to get these devices out of sort of laboratory testing and into devices in the market that we can use for our algorithms.
Okay, thank you very much. Thank you.
My pleasure.
Next question from Jorge Davila from Mexico.
Hello, Professor. Thank you for your amazing talk. I have a question about this insulin supply to the body. And we can consider that the control signal is a continuous signal to the body, or is in fact an impulsive one. It means that we have only access to the control in some specific periods of time.
Right. Okay. So first of all, Jorge, thank you for the kind comment. Can I ask what part of Mexico you're calling in from? Jorge, can you hear me? Oh, maybe he's muted again. I was just curious where he was calling from. So here's the idea. The pump is delivering what we call microboluses. So this is fundamentally different than a needle, which would be, as you say, impulsive, right? It would be just these impulses delivered 3, 4, 5, 6 times a day. But actually with a pump, they're very small microboluses that are delivered, and the delivery rates are modified every 5 minutes. So it really does begin to approach a continuum, but a continuum with now microboluses delivered with 5-minute intervals in between. So we're beginning to approach continuum, but at a fundamental level, there's still quantized delivery, but it's not the big impulses you pick up with meals.
Thank you.
Great. Thank you. Any other questions? I don't have any other hands raised at the moment. So, okay. Hernan de Batista, another question.
Just another small question, Frank. Could you please tell me how many children participate in your ongoing pediatric trial, and how old are they?
Yeah, so we're literally writing that paper up. Let me pull up the slide because I think I've got the count on there. This was the, here we go, the trial. So we had 48 children participate. Okay. And they were broken into these 3 age ranges. So one fraction were teenagers, 12 to 18. Another fraction were 8 to 12-year-olds. And then we had some very, very young children, 2 to 8-year-olds in this study. We wrapped this up right before COVID, mercifully. And so we're still finalizing the analysis and the write-up. We're hoping to submit this paper later this month. If you drop me an email, I'd be happy to send you a copy once we get through the review.
Okay, thanks.
Okay, Alejandra Ferreira de Loza, another question.
Yes, please. In the children case, do you consider a meal announcement? The children with the app in McDonald's or something like that?
Yeah, so in this protocol, the exercise was not announced. Some of the meals were announced. So we often do a mix. We'll often challenge the system with a meal that's not announced. But we have found, and maybe this goes back to the earlier question about glucagon and insulin and bihormonal, we find that announcing is really necessary when you're doing this outside the body. So when you are responding to a slow message, you're waiting for that glucose signal to come through the skin, and you're pushing insulin through the skin, you really need an advantage. So announcing the meals becomes critical in that respect. So in many of those cases, the meals were announced, but there were still huge, very complex, rich kind of fat, protein, carbohydrate profiles. So it was a very challenging problem with lots of snacks as well, and the snacks were unannounced.
Okay, thank you.
You're welcome.
Any other questions? Raise your hands. If I do have a question myself.
Please.
Thank you again very much for the interesting talk. I see that in a sense, the engineers rely on doctors to tell you that the patient has type 1 diabetes, right? So my question is a bit to the side. What happens if a patient is misdiagnosed and you attach one of these intelligent insulin pumps?
Yeah, so I would say the odds are low that would happen because we have very careful screening of these subjects coming into the trial. So for example, we will screen out a pregnant woman. We will screen out individuals who have serious heart complications. And so we also want to know what is their history with diabetes. How much insulin do they normally take? And do they have any residual insulin function in their pancreas? So that's all a part of the data we collect ahead to have a very careful scrutinous review before they could ever enter into one of our trials. So I've never seen a situation where somebody, in effect began producing their own insulin alongside our device. Now, if I were to hypothesize, I would say that things might not be so bad, because if they're making their own insulin, they're going to blunt glucose excursions, and our algorithm will respond to the glucose measurement. So our algorithm would reduce the insulin accordingly. So even in the extraordinarily unlikely case that we had somebody who had partial diabetes, I'd say there's no chance somebody could come through who doesn't have any form of type 1 diabetes. But if they had some functionality in their beta cells, I think our algorithm would still be robust.
Okay, so the following question is, then can you use your device to actually diagnose type 1 diabetes?
Yeah, I would say I see less possibility for that. Let me give you something maybe in the direction you're going here. Our solution is a therapy, and I should have said this at the beginning. It's not a cure. In fact, they wrote an article about our work a few years ago, referred to the care and the cure. The care is what I just talked about. The cure is something like stem cell or transplanted islets or something very significant that hasn't been solved yet. Where our device could help is if somebody is getting that kind of cure-like treatment, they're getting islets transplanted into their body, or maybe that stem cell therapy where they're in a partial state, or maybe these new cells are not quite fully up to speed, and you have the old manifestation of type 1 diabetes. Our algorithm on a pump could be the bridge that connects, going from the old state into some medical therapeutic state that is approaching a cure. But I don't know that this is the way that a doctor would use to do diagnosis. It probably wouldn't be the right way to do that. There are better ways to do that, I would say.
Okay, thank you very much. Any other questions? So, okay, we thank you again on behalf of the organizing committee, on behalf of the Association of Automatic Control. We thank you very much for this interesting talk. And we wish you all the best in this new, okay, as you said before, when you were talking with Ricardo, you have elections in the US shortly, so let's hope for the best.
I appreciate that. Let me say once again, it's a regret that I can't actually come to Buenos Aires or some other part of Argentina to deliver the talk. I'll save that for a future trip. But it's been my great honor to talk with you, and I'll say that I always measure these talks in terms of the quality of the questions. And there have been some really thoughtful, insightful, very interesting questions. So thank you for those.
Okay, thank you very much, and goodbye.
All right. Take care. Stay
Goodbye.
safe.
Thank you for your answer. Thank you.
Thank you.
Goodbye.
Bye now.